Yeah, you have a cricket assignment. Okay, so we left off talking about the last of the lipids, which is the steroid hormones, right? And so you guys know that all of these are synthesized from cholesterol. And we talked a little bit about cholesterol's bad, but without cholesterol, you wouldn't be able to synthesize testosterone or estrogen or cortisol or any of the hormones that you require for life. So without that, you wouldn't be alive. So you need some amount of cholesterol. And in fact, your body synthesizes cholesterol when you're low on it, and usually it will get rid of it if you're high on it. So most people, it doesn't matter what you eat. 80% of the population, your cholesterol levels won't vary based on what you eat. Only 20% of the people that affects. So cholesterol is the basis for all of these. This is cholesterol. This is a molecule called 7-ketocholesterol. Why do you think it's called that? That's a ketone. What carbon do you think it's on? There you go. And then testosterone and estrogen. So you can see they all are basically synthesized off of the same molecule, cholesterol. This actually increases your risk of cardiovascular disease. This is one of the chemicals that we found does. This does not. Alright, so we're going to move on to the proteins. You guys know that proteins, probably from the chemistry lab, or maybe you don't remember, but proteins are made out of what? Amino acids, right? And we talked a little bit about amino acids when we did the functional groups. So amino acids are named that because they have an amino group. What's an amino group look like? Good. And then it has a carboxylic acid group. And so this is where it gets its name, amino acid, right? And then we have this R, which varies, just like DNA, right? How many letters in DNA? 4. It's A's and G's and C's and T's. This has 20. There's 20 different amino acids, right? So how many letters in the alphabet? 26, right? So it's just a lot more variation that you can put in there to build what you want, right? Right. So proteins, when we take 2 amino acids and we link those together, we call that bond a peptide bond. Just like when we took 2 sugars together and we linked it together, what was that bond called? It's called a glycosidic bond, right? So in between amino acids, it's called a peptide bond. And because of that, sometimes proteins are referred to as peptides or polypeptides. That means the same thing. Right? So monomers are amino acids. We put these in long chains and we make proteins. Proteins are what you are, right? We talked about this. So the central dogma is DNA is made into RNA, and RNA is turned into protein. And protein is literally what we are made out of. So just to give you some examples, keratin gives us structural support. So you guys know what keratin is? Principal component of your nails, hair, right? And you may not know this, but it actually holds the nuclei shape, so it allows the nucleus to be round. We store amino acids, right? In our blood, we store protein in the form of albumin, which is another protein. We use it for transportation. So hemoglobin is a protein, right? What's that for? right? And it binds oxygen to carry oxygen to your cells. And then it's used for signaling, right? So insulin is a protein. The receptors in your eyes are made out of proteins. That allows you to see and see in color. Movement, you wouldn't be able to move if it wasn't for myosin, which is a motor protein found in your muscles. And I showed you guys the pictures of the myostatin, cows and stuff. And then your immune system, right? Your antibodies are proteins. So they're chains of amino acids linked together. And then lastly, and probably most importantly, is that enzymes are the workers inside your body, and they are made out of proteins. Without them, you wouldn't be able to do any of the chemical reactions that you need to, say, digest your food, or build molecules, or break them down, or carry oxygen, or anything like that. So enzymes act as catalysts. And we'll talk more about this, what a catalyst is, when we get to chapter 8. So any questions about that? What proteins are, what the bonds are called? Okay, so you should know amino acids. You should be able to recognize that. If I drew this on the test, you should be able to tell me that that's an amino acid. You should also be able to tell me that that is an amino group, that's a carboxylic acid group. You should be able to tell me that's a hydrogen and that's a carbon. That was a joke. Trying to lighten it up a little bit. You guys look a little serious out there. Must be getting closer to the test, because I can tell students start just quietening up, no questions, freaking out. So it's a 1 week from today, 2nd. Yeah. So peptide bonds, they're formed just like all other macromolecules. So if we want to put 2 amino acids together and form a peptide bond, what's the reaction that we have to do? Dehydration. We take water out. And whenever you eat proteins, right, what is the reaction that breaks them down that allows you to digest them? Hydrolysis. Excellent. So proteins are the same, right? This is always the same. Every amino acid has the same amino, carbon, hydrogen, and carboxylic acid. So when we link these together, that repetitive part that's never varies, we call that the backbone, which is shown right here. And the ones that vary, the parts that vary, are the R groups, right? We call those the side chains. Everybody good with that?
So like on this diagram right here, where it says the peptide bond right there.
Right here, yep.
So that's where.
So this is also important. There's an amino end. Then we have lots of peptide bonds. There's a peptide bond. That's another one. And we have carboxylin. So no matter how long we make this, right, there's always going to be an amino group on one end of the train, right, like a caboose, and then there's going to be an engine on the other. So amino group is always on one end. Carboxylic acid is always on the other, no matter how big we make this. And that's important.
What is it bonding with? What kind of group is that? The other amino group?
Where?
Like, you know, where the amino is bonding. What's that group?
This?
No.
Here?
Yeah. The whole
That's a carboxylic acid. So it's this group. The water was removed. The OH is removed. And then that's linked to the nitrogen in the next chain. So the hydrogen off of here combines with the OH, and that bond is formed.
And those are 2 amino acid groups right there?
Yeah, so this is 1 amino acid, that's 2, and this is a third that's coming on. So this is 3. So we would call this a polypeptide because it has more than 1 peptide bond in it. What's shaded here in purple is the backbone, the repetitive part of it. Right? DNA has a backbone too, right? Because the sugar is always the same. It's always deoxyribose. The phosphates are always the same. What varies? It's the bases, the A's and the G's and the C's and the T's. So This is pretty typical of macromolecules.
Does it have to be the same structure?
What do you mean?
I've always going to see exactly the same here as
You mean the type of amino acids? Yeah, no, you could put anything, right? Like, it's like if I had an alphabet, I could put A next to a B or A next to a C. You guys don't know the alphabet of amino acids, and I don't expect you to know that, but there's 20 different kinds. And I can put them in any order I want, right? I could put tyrosine next to threonine, next to arginine, next to leucine, next to isoleucine.
Does that happen?
I can put it in any order I want. Sure it does. That's how proteins are built. So, I mean, the way that our groups interact with each other, these proteins, the way that they function is they have a specific shape. That's the most important thing in biology is shape. So, if I have another, so this is how it would end up. So let's just say that this is arginine, and you guys built these, and let's say that this is leucine, right? So let's say that these two have charges that are positive. They're not, but let's just pretend. So these two positive charges, what are they going to do? So it's going to bend this whole protein, right? What if that were negative? What would happen? So it would bend the other way. And that would make a different shape, wouldn't it?
Right.
Or this could be neutral. It could be like hydrophobic. And so what would happen then? Yeah, it would just be a straight chain. So and then, you could have this part bend this way, and eventually you're going to get the fold into a specific shape. And that's going to dictate its function. Well, that kind of leads us into what we need to talk about next, which is the three-dimensional shape that proteins end up becoming. So the sequence of amino acids determines the shape of the protein, and that's found in the DNA and the genes. The way that we look at this is that you have these different interactions with hydrogen bonds and ionic bonds and R groups and stuff like that, and it causes them to fold into a specific shape. This is what we call a space filling. So that shows the atoms and the electrons, right, taking up space. And you can see that. So this is what it would look like if you could shrink yourself down, right? It has a specific shape. It's hard to see the interactions inside this, right? So scientists often use what we call a ribbon diagram, which is what this is. And from this, I can see certain things, like you can see that this is a helix, right? It's like a ribbon that's been twisted, and these are flat. Everybody see that? Well, these are all. This is just chains of amino acids linked together, right? So proteins are big. Usually they're anywhere between 100 and 1,000 amino acids. So they're massive. I mean, you built this, and you built it with 2 amino acids. Now imagine building one with 100 or 1,000. And you can see that they get really big really quickly. So these shapes right here, these helices and sheets, are formed by the interactions of hydrogen bonds in the backbone itself. I'll show you that in a second. What do you think these yellow things are? They are bonds. What kind of bonds? No? No, they're yellow for a reason. What's yellow? What'd you say? That's right. You guys in chapter 4, you learned about disulfide bonds that help make proteins hold their shape. So from this, I can tell, because there's quite a few of these, that this is probably a protein that's not inside a cell. It's probably outside in the blood, moving around, maybe in the eye or in the mouth or the stomach or something like that. Does that make sense? What? That represents a disulfide bond. Just like this represents the letter A. That makes sense? So this is a representation for scientists to look at and try to figure out like what's going on. I mean, trust me, pharmaceutical companies are looking at the shapes of these things because if you have a receptor on a cell that when it binds something it causes pain, what would you like to do? If you want to stop pain, block the receptor. How are you going to block the receptor? You got to find something that fits in it. And so if you don't know what's going to fit in there, then it would be like you going to an auto parts store and just buying random stuff to repair your car.
So the bond between the hydro right there is an ionic bond?
No, those are covalent bonds. They're strong bonds. Disulfide bonds. We talked about this in chapter 4 of the functional groups. Remember? Alright, don't worry about it.
I'm trying to think, like, where would it bind with that? Because it's SH.
It's here. It's in these, right? So some of these R groups have sulfurs in them, and they interact with each other. Okay? So this are just long chains of amino acids. It's just representing the shape that it takes on. Hopefully it'll make more sense when we move on to the next slides. So here's a good example. This is DNA, right? This is the enzyme that copies DNA. So do you think that the enzyme that copies DNA would work if it didn't fit, if it couldn't read the DNA? Could you photocopy your book if it wouldn't fit on the platen of the photocopier? So, it's really important that the shape of these molecules fit whatever their function is. So here's the 4 levels of organization that we talk about in biological systems. So we have primary, secondary, tertiary, and quaternary. Quaternary, all proteins don't have a quaternary structure. It's only if you need 2 or more to make a specific shape. Does that make sense? So like, here's one marker, right? This wouldn't have a quaternary structure because it functions on its own. But if the protein needed 2 markers and had to have that shape, then they would have a quaternary structure. Does that make sense? So some proteins are like that. You guys are just staring at me. What? So if it has more than one protein needs to come together to make it functional, then it has a quaternary structure. Think about it as a car. Could your car run on 1 tire? No. So it has a quaternary structure because it requires 4 subunits of tires. But if you were riding a unicycle, it wouldn't have a quaternary structure because you only need 1 tire, right?
But they'll all have a tertiary.
Yes, everything has a primary, secondary, and tertiary. So I'll show you in a second. Hopefully this will make more sense. Alright, so you guys have written words before, right? And you know that the order of the letters are important in determining what word you're making. Same with this. So the linear structure of proteins is determined by the order of the letters in the DNA. Because the DNA is turned into RNA, right? And the RNA is read into proteins. So this particular protein, we number the proteins from the amino end to the carboxyl end. So this would be number 1. These are the different amino acids, right? I don't want you guys to memorize these. I just want you to know how this is numbered, because this is how scientists talk about diseases. So this is lysine, valine, phenylalanine, glycine, arginine, cysteine, glutamine, leucine, alanine, so on, methionine, lysine, histamine, glycine, and so on, aspartic acid, blah, blah. So all these are just orders, right? They're just letters. Does that make sense? Then there's 20 different kinds. So we number from here, and if there's a mutation that causes a disease, then we represent that by the number, the location of where the amino acid change has occurred
So like
to make the proteins change its shape.
Benadryl is an antihistamine. So does that mean that it bonds on the histamine molecule?
No,
Blocks the receptors on it?
not really. So we'll go down this tangent. So histamine is from mast cells, right? There are certain cells that acquire and accumulate histamine. So when your body perceives that it's getting some sort of infection or something like that, it will release histamine. What histamine does is it binds to, and it's a vasodilator, so it opens up your blood vessels to allow white blood cells to travel out so it can go to the site of infection. But if you get too much, it makes your blood vessels too leaky, you can have a massive drop in blood pressure, and that could cause massive problems. So an antihistamine prevents the mast cells from releasing their payload, so to speak. So in a way, yes. Yeah, but not directly. It binds to the cells. Make sense? Well, not all antihistamines work the same way, so that's probably not true. There are probably some other classes of drugs that work differently. But
Oh, I want to know if that's where it goes. I noticed histamine and that.
Okay.
My brain thinking out loud, sorry.
Okay, all right. So here are these are normal red blood cells. This is hemoglobin. This is the amino acid sequence of part of the hemoglobin gene. Hemoglobin is, roughly 130 amino acids, depending on what species. Here you can see this is sickle cell anemia, so the cells are sickled, and this is caused by a single mutation in number 6, right? So if you were talking to a scientist, you wouldn't want to go, okay, it's number 6 and start counting from here, because that would be completely wrong, right? You always start from the amino end. So we have a glutamine mutated to a valine, and that causes sickle cell anemia. So that one single change in that one amino acid causes the hemoglobin to change its shape. It can't load oxygen, and so the cell collapses on itself. It's kind of like a balloon being deflated. And this causes the cells to not be able to carry oxygen, so you're anemic, but it also causes the cells to sort of hook into your blood vessels like little sickles, so that's pretty painful. And then it can clog up your spleen that filters your blood, and you can, it can cause your arteries to clog, so you could have strokes, and all kinds of nasty things come from this. So it's not a pleasant disease to have at all. Any questions about primary structure or histamine? All right, so secondary structure is the structure, the shape that's determined by the interaction of the backbone of the amino and the carboxyl groups. You guys know that, well, you should know that nitrogen and hydrogen, they don't share equally. Remember from chapter 3 we talked about this? So nitrogen keeps the electrons, so this builds a slight positive charge on hydrogen, slight negative charge on nitrogen. Same thing here, so, and here, actually. So we have a slight negative charge on oxygen and a slight positive charge on hydrogen. And so that causes attractions. And so you can see that these little dots are showing the hydrogen bonds. So remember the helix that I showed you from the ribbon diagram before? So this is called an alpha helix, and it's formed by hydrogen bonds in the backbone. So on the test, I might ask you, what causes secondary structure? Hydrogen bonds where? R groups? Right. And then what? So there we have the alpha helix, and then the other one is called beta sheets, which is right here in the backbone. So right here, hydrogen bonds along the backbone. This is the backbone right here. So amino, carbon, carboxyl, amino, carbon, carboxyl, amino, carbon, carboxyl. This is the R of the variable groups. There you go. So I might ask you on the test, what are the two kinds of secondary structures that are formed in proteins? Alpha helix and beta sheet. Okay, now I ask you another question. Which one is stronger? So these are the same number of amino acids in this alpha helix and this beta sheet. Which one do you think is stronger and why? So which one do you think is stronger? The one that has more hydrogen bonds or the one that has less hydrogen bonds? So then what's stronger? The beta sheets, right? Yeah, because they have more hydrogen bonds, so they're stronger. In fact, these are the principal component in spider silk, which is about 10 to 100 times stronger than steel by weight. In fact, it's stronger than Kevlar. So what would be some uses of spider silk? Black jackets, helmets. One of my students said that they're using spider silk for screws in surgery for like plates and yeah, in fact they never have to be replaced again. Yeah, you have to have what's called revisions whenever you have like partial replacements or total replacements. What else?
I watched something this company out there with suits that are just by the fabric.
Right?
So this weighs just like a normal piece of clothing, but it's
Yeah, so you could make that out of spider silk. It's be stronger than Kevlar. What about well, yesterday I saw a thing on television that the Japanese want to build a space elevator. Space elevator.
they just slingshot you out there, pull you back with a spider.
Yeah, so it's not hard. I mean, if DirecTV can put satellites in a geosynchronous orbit, I think NASA could.
Yeah, but if it's moving, the location on Earth would have to move.
Right. So geosynchronous means that it rotates at the same speed as the Earth. So you have the Earth here, right? You send up a satellite, and the satellite rotates at the exact same speed that the Earth does. So it all seems like it's stationary. Right. That's why you never have to adjust. If the Earth moved in like your DirecTV or whatever satellites didn't move with it, you'd have to adjust your dish to point to the satellite all the time.
How would you stabilize that?
So what do you mean?
Stabilize it on Earth because the gravity is gonna lessen as you get higher.
Right.
You have to stabilize it down here so it doesn't.
What if a bird flies into it?
It's gonna be stronger than a bird, but just thinking because steel is gonna bend with motion and it's gonna produce.
And what is the point of this?
Well,
Why are we doing this?
Well, right now, I mean, sending a payload into space costs hundreds of thousands of dollars per pound. But with this, it'd only be like 10 or $100 a pound. That means that you wouldn't have to have like a boy band salary to go into outer space. You could, pay $300 or $400 and ride up into space on a space elevator. Why not?
How long is it gonna take to get to space?
I don't know. But the problem is, how are you going to build a cable? What are you going to build a cable out of?
Spider silk. Totally don't trust it.
Spider silk. What? I mean, you couldn't make it out of steel. What if it broke? It just come crash 600 miles or
Spider webs.
1,000 miles of cable. Yeah, but I mean, what if a spider web broke and fell on you?
I know, I'm just saying, like, you're making it out of spider silk and spider webs break.
Yeah, but they're stronger than steel. Yeah, so that's the question. Like, right? You can't.
And
Spiders.
is one spider silk stronger than the other?
Oh yeah. Spider silk is the second strongest substance known to
But I mean,
man.
is one stronger than the other? Like, yeah.
Like a black widow and a tarantula.
Which one's stronger?
So the strongest one comes from the Madagascar golden orb spiders.
Those are the ones that build the real pretty.
Yeah.
Then are those ones that are like big enough to touch a human? Is that
I think so, yeah. Because they're almost like synchronized, like how the distance and everything's perfect.
So these orb spiders, right? You can't just really say, okay, I want you to build me a space elevator, because they're not like silkworms. They don't just spin webs. Is it seeing things like a laser light? Never mind. Probably looking into the microscopes too long. Anyway, so you can't just tell them to spin these webs, and so how long would it take? How many spiders would you need? They've made other stuff.
They do spin webs pretty quickly.
They've made other stuff out of spiders, like
I think the stickiness of it.
so. Here's a scarf that was made out of spider silk. How many spiders did it take?
a lot?
How many years did it take?
That's me. I'd be so mad if I was a spider.
So I mean, that's not anywhere near the size of what we need for a cable.
20 years with like almost a population of 30 million spiders.
Probably more than that. You're,
Build at least what?
right. So is
30
it
miles of spider?
so is it possible, or is it just a pipe dream? Oh, come on. You guys don't give up on me.
It's possible.
What makes spider silk?
Spider.
Okay, rudimentarily, yes, but on a more molecular level.
Amino acids.
Proteins, right? You know, I already showed you, you can take any gene from any organism and put it in any other organism.
What organism would you put it as? That would just
Oh, I'm
shoot
glad
out
that
spider
you
webs
asked that.
out of its ass? Just milk it out of a cow?
Spider goats.
You're pretty close.
Oh my god.
Goats?
Goats.
We really did that.
God.
So scientists have taken
5 seconds.
the gene from the golden orb spider that makes silk and put it into the cells that line the mammary glands of goats. And so when they make milk, they make the protein for spider silk.
How is it the same strength?
Yep. And they centrifuge it out. Yeah, it's exactly the same.
I know. I'm like, wait a minute.
Well,
You're
no,
squirting
no,
web
it's
out of there?
so the way spider silk proteins are laid up on top of each other, so they're in small portions, and you could just spin them in a centrifuge and get them to reassemble like that. So it doesn't. You can't tell the
Don't
difference.
cows make more milk?
I don't know. I didn't do it, right? You gotta ask the people that did it. But you can go visit them. They live in Wyoming. Maybe for Halloween.
So what are they planning to produce that elevator out of these goats?
He's describing a way that we
It could happen.
proteins faster. Just harvesting spiders.
Yeah, that's exactly right. You never know. But it's possible, isn't it?
So, but those goats definitely can't give out offsprings because they.
Did that render it sterile as well?
No.
No, but
They can have baby spider
How would
goats.
it feed its baby?
And so
The milk isn't tainted. It just has proteins are just amino acids, right? Goats drink milk and they break down into amino acids just like you. So it's not. I mean, you could eat spider silk if you could break down the proteins in
I'm
it.
thinking the cotton candies coming out of these goats.
Yeah.
I'm really thinking like cotton candies coming out of these goats.
It's not like that at all. It looks like normal milk. It looks like normal milk. Okay. All right. So anyway,
Wait, so now.
something to think about.
If you take a regular baby goat and you give it to the spider goat and it drinks the milk, does it change it?
No.
Okay.
It's like that's like me asking you if you ate a loaf of bread, would it change you into bread? No. Yeah, it doesn't affect it at all. It's just milk,
But
you
if
know.
it's born from
Milk has
the
proteins in
spider
it
goat,
already.
Spider goat.
Yeah, because it would have the gene that makes the spider silk.
There's no like deformities from this.
No, the only difference is, do those goats look deformed to you? No, they're normal goats. They make goat noises and play in the goat fields and make goat babies and make goat milk. Nothing different. They don't have like 8 arms or anything. They're not crawling around making webs. Let's move on. So the next level of organization for proteins that give them their shape is the tertiary structure. This is caused by the interaction of the R groups, right? So on the test, I might ask you, what causes secondary structure to be formed? Hydrogen bonds where? In the backbone, right? And tertiary structure, it's caused by R groups, right? And so I'm not going to make you memorize this, but we talked about it could be positive or negative, right? So it could be ionic bonds, it could be hydrogen bonds, it could be hydrophobic or hydrophilic, and we'll talk about this later. But let's say you have a protein and you want an enzyme and you want to bring water in to cause a reaction to occur. What kind of R groups would you want in there? Hydrophobic or hydrophilic? Hydrophilic, right? So guess what? The enzymes that perform reactions that put water in to break molecules, what's that reaction called? You take water out to build it. No. We add water in.
Oh, that's hydrolysis.
Yeah. So enzymes that perform hydrolysis are ones that have R groups that are hydrophilic at that point where the reaction occurs. That makes sense? So that's how it works. If it didn't have that, if let's had hydrophobic groups, would you be able to break down your food? No, and then that would cause either you to have a disease or die. So the order of these groups are really important. And then quaternary structure, right? So we talked about that. So hemoglobin, there's a gene for hemoglobin A and a gene for hemoglobin B. So these make individual proteins, but your hemoglobin requires 4 parts, 2 alphas and 2 betas to make it functional. So that is quaternary structure, right? If it worked by itself, if you just needed 1 alpha hemoglobin, it wouldn't have a quaternary structure. Does that make sense? By the way, a heme is something that binds a metal, a protein that binds a metal. So what metal do you think hemoglobin binds? That's where it gets its name. Iron. That's why blood's red. So here's 2 beta and 2 alpha chains of hemoglobin, and all together they make a functional hemoglobin molecule. So that's what it looks like. Same thing with collagen, right? Stuff that people inject into their faces. So this has got 3 strands in it, but each one of those is made as an individual protein. So it has quaternary structure because it won't function as a single strand. Does that make sense to everyone?
How do we know which one it's in? So if we were to look at
Oh, I told you. You'd have to study it. You'd have to find out what the shape is that it makes. All right, so this is in your text, and it's just an overview of what we just talked about. So primary structure is what?
linear.
It's just the order of the amino acids, right? And that's determined by what? What determines the order of the amino acids? The DNA. Exactly. The order of the nucleotides in the DNA. All right, so then what determines secondary structure?
Hydrogen bonds in the backbone.
In the backbone, right? There's 2 different kinds. What are they?
Alpha and beta.
Alpha helix, beta sheet. Which one's stronger? Good. And then tertiary structure, what forms that? Good. And then quaternary structure is when 2 or more proteins come together and make a functional one. If you don't have 2 or more, there's no quaternary structure. Does that make sense? All right. So what happens if I change the primary structure of okay, so let's go back to this example. Let's say that these are both positive, and that's normal. Let's say that they get a mutation, and this changes to isoleucine, which is also positive. It's not really, but let's just pretend. So these are both positive. Will that change the shape of the protein?
No, it's still positive.
It was already positive, right? So you could have a change that wouldn't affect it, right? But most of the time you would have a change that would affect it.
Is it just the positive or the charge of the R groups that causes the shape?
No, it could be that they're hydrophobic or hydrophilic. It could be that they're acidic or basic. In fact, in your book, it lists all the different amino acids.
So technically, if the other one that you put on, other group that you put on there is different, it could change the shape. So it could, even if it's still positive, it could change the shape.
So here's the question. Well, it probably wouldn't if it's the same, right? It wouldn't change the shape because it's chemically similar. Yeah, but they already were. You had a positive up here first, and you switched it with another positive. What did that do? Nothing.
But if it was hydrophobic,
Well, there's only 4 classes, so I'm telling you it's a positive, not a hydrophobic, not a hydrophilic. It's positive. There's 6 classes of amino acids. You guys are making this way harder than it has to be. Just follow me, all right? Let's forget the names.
just said
I have a positive, I switch it. There's a positive, I switch it with another positive. What will it do?
nothing.
Right. Does it change the primary structure? I just told you I changed one amino acid with another. That's like me changing one letter with another. Does it change it?
Yes. Yes.
Yeah. Does it change the secondary structure?
No.
No. Tertiary structure?
No.
No, it's the same, right? Because I changed it with one that's chemically similar. So, having a mutation of an amino acid will definitely change the primary. It may change the secondary. It may change the tertiary, but it doesn't have to because that substitution could be something that's chemically similar. Right. So one class of the amino acids are defined. They're all the same, besides the R group. So changing one you would have to change the R group. You wouldn't change anything else because the backbones are the same for every single one. Ooh, okay. Exhausting. Next, nucleic acids. So you guys know what these are, right? What? DNA and RNA. We talked about this. What's the difference between DNA and RNA? You're close. So it's ribose, and deoxyribose is the sugar.
One oxygen.
It is one oxygen. Where is that oxygen located?
Two.
Yeah, carbon 2. Alright, so this is ribose. The carbons are numbered in clockwise from oxygen. So the bases are located on the number 1 carbon. They can be an A, a G, a C, or a T. In RNA, the T is replaced with U. So in DNA, it's AGCT. In RNA, it's AGCU. Got it? Both of them have 4 letters. This is, what sugar is that? No. Yes. This is ribose. And what is that? Deoxy, right? It took away the oxygen. That's the only difference. So in DNA, this is the backbone. It's the same, right? Over here, this is a continuation. So this would be another letter up here. That's the number 2 carbon. So I'm cheating. That's the 1 carbon. That's the 3. And over here, this would be the phosphate. And then we have another base over here, right, which would be different. So let's say that's a C, and let's say this is a T, right? So this right here is the backbone. It's made of the same stuff. Deoxyribose, 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 deoxyribose. Phosphate, 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 phosphate. So this is considered the backbone of DNA, just like in the proteins, right? And then this, it's kind of like the side chains, except here, these are bases, DNA bases, or RNA bases. DNA is AGCT, and RNA is AGC. Do I get it?
So like how would like the protein backbones like starts with the amine and then with the carboxyl group. Here it starts with phosphate and then with the deoxyribose.
Right, but when we number DNA, we say it's 5' to 3', or DNA runs anti-parallel, so the other side would be 5 to 3 in the opposite direction. What's the difference between those? Yeah, so I'm going to show you that in a second.
So the difference is the oxygen.
Well, no, he's asking about the bases. These are different chemical structures. The difference between DNA and RNA is that presence or absence of that oxygen. which is shown in this slide right here. So this is deoxyribose. It has one less oxygen. This is ribose. It has an OH here. That's the difference. Alright. So you guys know DNA is found in the nucleus. DNA is just an instruction book. It doesn't really perform anything. Its job is to tell the ribosomes, which are what are found in your cells, what kind of proteins to make, and it does that by telling them the order of the amino acids to build. Does that make sense? So there's three types of RNA. So I just remind you, DNA, right, is turned into RNA, and that's turned into protein. And this process, because there's 4 letters in DNA and there's 4 letters in RNA, this is called transcription. Because it's just transcribing it from one form to the other. It's not changing it. This is going from 4 letters to how many letters in proteins? How many R groups? Yes. So this is like going from one language to another, right? So what do you call it if you're taking something and converting it into a different language? So that process is called translation. This is done by an enzyme called RNA polymerase, because it's making a polymer of RNA from DNA. And this that does this is called a ribosome. So in a ribosome, it has to feed the RNA inside of it. And the RNA has a 5' and 3' direction too, just like DNA. So in here is something called ribosomal RNA, and that lines it up and tells it what direction to read. Right, so if you're reading in English, how do you read? Don't read right to left. In English? Wrong one, yeah. So in Arabic, it's the opposite, right? But you need to know what language you're reading in order to read it correctly, right? If you read rat backwards, it wouldn't mean the same thing, would it? It would mean tar. So this helps line up this message so it gets read in the right direction. Because if you read it in the wrong direction, you're going to make the wrong protein. And that would be bad, right? What if it's hemoglobin? So ribosomal RNA does that. Transfer RNA, tRNA, brings in the amino acids, the right amino acids, and translates that code from this is the messenger RNA. This is the one that came out of that's this one. Came out of the nucleus. Okay? So tRNA reads the message and puts in the right amino acid. So like we were talking about for say it's reading this, right? So the tRNA would read the messenger RNA from DNA, and it would say, okay, that first one is a valine, because that's what the code in the messenger RNA is. Because the letters are different, right? So you need somebody to translate it. The second one is histine, and so histidine, and so on.
So the tRNA reads from DNA, the RNA is taking on nucleus.
Right. So, okay. So I'm going to go to DNA.org because I think that you guys can see this visually better than I can explain it. There's a link for this site on the web links and Canvas too. So I'm just going to click on the code. And I'm going to click on copying the code and putting it together, and I'm this is how transcription works.
He is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein.
So we're looking at this right here.
Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands.
So that blue molecule is RNA polymerase. It looks at the DNA and it makes a copy of every letter as it goes along. So here's the double helix of DNA, the red right here, or pink or whatever. You can see it's opening it up and reading it, and then out is coming out the RNA. So DNA is double-stranded and RNA is single-stranded.
Strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA.
So that specifically is mRNA, or messenger RNA. This kind of RNA.
The yellow strand.
Yeah.
The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
But it doesn't have those sound effects, I promise you. Alright, so that is going from here to here, right? So now I'll show you what happens from here to here in translation. Yeah.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the right.
These are transfer RNA, the green triangles. The little red tips are the amino acids.
Ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside
What caused it to fold up in that shape? All right. So, and then this, you can pretend like you're a transfer RNAs, you're a ribosome, and so you direct the conversion of messenger RNA into amino acid sequence. So here's the messenger RNA. What would match to this? You guys know? Remember A? So remember, there's no T's in RNA. U, A, C. Good. So UAC. That is correct. And then it would move over 3 letters. And then the next one would be good. So this is serine, the amino acid serine. The one before that was methionine. You guys don't like to know that, but that's what the letters stand for. Different R groups, right? Different chemicals. And then what's next? U.
So what happens to the messenger? Those are ribosomes? No, not messenger triangles.
The transfer RNAs.
Transfer RNAs.
So they deliver their payload. They're like a UPS truck. They deliver what they want, and then they go back to the home base, and they get a new amino acid, and they come back in.
So they do keep doing it. They don't.
Your body is really good at recycling everything. It doesn't want to waste any energy. It's very efficient.
So the home base would be.
It's an enzyme called amino acyl tRNA synthetase. We'll learn this in chapter 17, right? So don't get yourself overwhelmed, right? Focus on this chapter. The what? To make proteins. Your genes are in your DNA, right? Those are made of A, G, C's, and T's. But that's not what makes you what you are. What makes you what you are the proteins. So you have to convert this system into the protein, the 20 amino acid protein system. So how do you do that? This is how.
Okay, so DNA is separated, right, from the double helix to the single helix. One side is replicated and the other side is turned into.
Not replicated, it's transcribed.
But only one side.
Right.
What happens to the other side?
Well, nothing. A gene is only on one side of the DNA. You could read it in the opposite direction, but it would be different. We'll talk about this when we get to chapter 17. Right? But you're right. Only one strand is read.
That's what I meant.
Yes, that's correct.
And we're not going to talk about what happens to the other one here.
The other strand doesn't do anything. If it's not made into messenger RNA, if it's not made into that, it doesn't ever get made into a protein.
So it just looks good.
No, it's there's a reason that DNA is a double helix. It's more stable. It protects itself, right?
So on Monday, we're just going to review everything on Monday, or we're just...
I'm not coming in on Monday.
I mean, sorry, Tuesday.
Yeah, we'll do a
We're trying
review.
to get through chapter 5 right now.
I'm trying to get through chapter 5, but I don't think it's going to happen.
I don't mind staying late and finishing.
All right. So DNA is double-stranded, and that double-strandedness helps make it more stable. You want your DNA stable, right? And you probably know DNA stable. Like, they're digging DNA out of mammoths, right? They found it in a Tyrannosaurus Rex femur that's 125 million years old. So DNA can last a long time. RNA is inherently unstable. That's why it's single-stranded, because it's easily broken down. I mean, what's the difference between the two? One's double-stranded and one's single-stranded. Besides the nucleotides, the A's and the G's and the C's and the T's and the U's are found in, T's are found in And another difference is that A's pair with T's in DNA, that has 2 hydrogen bonds. G's pair with C's, and that has 3 hydrogen bonds. And then remember, DNA is anti-parallel. So remember, what carbon is this? This is oxygen, so that's number 1 with the base on it that I drew up there. Number 2, number 3, number 4, 5. So this is going 5 to 3, right? And this one's 5 up here, and this is 3. So this strand on the left is going 5 prime to 3 prime, and this one over here is going 5 prime to 3 prime. So they're flipped around, right? And we call that anti-parallel. That's just the name of it. Okay, so remember we talked about, so the monomers that make up amylose are what? The monomers that make amylose are glucose. What are the monomers that make triglycerides? Glycerol and fatty acids. What are the monomers that make proteins? Amino acids. What are the monomers that make DNA? They're called nucleotides. So this is a nucleotide right here. It's made up of the pentose sugar. So if this were DNA, what sugar would this be? DNA. Deoxyribose, correct. If it was RNA, it would be ribose. Good. This is the base. Remember, it's attached to the 1 carbon. This is what makes the letters, the A's and the G's and the C's and the T's and the U's. This is what the bases look like, right? So this is T. It's closely related to U. What's the difference between these two? What's that called? No. What's a C with 4 hydrogens on it called? Methane, right? So this is a methyl group. So T's have a methyl where U's don't. They're missing it. But otherwise, they're exactly the same. And what do these all have in common? C's, T's, and U's. They're a single ring, like this. These A's and G's, they have 2 rings like these. So ones with single rings we call pyrimidines. Ones with 2 rings we call purines.
The reason the A's are T's is because they got hydrogen that they.
No, it's hydrogen bonds, right? So it's just held together by interactions between the nitrogens, oxygens, and hydrogens. In H2 where? No, so these don't join, right? They're held together with hydrogen bonds because we want them to be weak. We want them to be able to open up. So if you're not sure, go back and look in your lab manual because it shows you the interactions of that in that when you did the chemistry models, which you probably forgot because we did that like the second week. Okay, so a nucleotide is made up of a base, a pentose sugar, and a phosphate group. So this is the backbone because it never changes, right? This is like the side chain because it contains each of the nucleotides. So for the test, I don't want you guys to memorize, these chemical structures, but I do want you to know which ones are the pyrimidines and which ones are the purines. Right, but also I might ask you on the test, what is G? Is that a purine or a pyrimidine? Yeah, because you could read it on the slide, but you need the other know. Yeah, no, that's cool. So you make sure that you guys know that, because that's important. They have different shapes, they have different chemical and physical structures, and we're going to talk about how your body detects DNA damage and DNA repair, and it has to do with shape. And if you don't understand the shapes, you're not going to understand the stuff that we're going to talk about in the future. So it's important that you know this. Not that you know each one of the chemical bonds in there, but the general shape, right? One ring versus two rings, and what they're called, because that's the terms that scientists use. I'm not trying to be mean. I'm just trying to teach you guys this. So on the test, I might ask you, what are the building blocks of DNA? What are they called? Nucleotides. What's the building blocks of RNA called? There you go, nucleotides. Good. Make sure you guys know the difference between the two, right? You want your RNA to be unstable because we talked about this. If you went outside and it's cold and you started shivering, that's done by RNA making proteins that cause you to shiver. So if it was stable and you came back inside and you got warm, you would still shiver, and that would be lame. So you want RNA to be unstable. You want your DNA to be stable because you don't want mutations to affect everything that's made from that. All right. How much time do we have left?
Keep going.
Yeah, we're good. We're almost there. There's like 3 slides left.
All right. So
Nobody's packing anything.
this isn't. I can cover this later.
3 slides.
We want to review on Monday.
Yeah.
Well, I can do the 3 slides on Monday, and then we can just do a review. Tuesday. Yeah, I'm not coming on Monday. Did I say Monday? You made me do that, didn't you? Yeah, we'll just pick this up. I don't want to rush through this last part. We'll cover this on Tuesday. We'll finish the last 3 slides, and then we'll do a review for the test on Thursday.
We were trying to get you to say, recording here. That you'd be here Monday.
No, I'm not coming on Monday.
So, are you going to be here Tuesday for class? Like around 5:30? Say.
On Tuesday, yeah. So, because I do special projects with these guys, so
So can I bring my list of questions?
I'll be in the undergrad lab at 7 o'clock. The undergrad research lab at 7 o'clock.
7 o'clock? Not 5:30?
No, I teach in the other campus.
Can we do one example of just right now so I can see?
No,
Please go over
it's 11 o'clock. It's 10 at night. I want to go home.
5 minutes.
I haven't seen my kids all day.
Yeah.